So we are going to finish Unit 7 from this lesson. And we discussed already what is the principles of breeding technique. So we are trying to change the natural selection to improve the quality, quantity, and other factors of the organisms. And we uh, we can use different principles when we use breeding techniques. So far, what we discussed, uh, kind of inbreeding, outbreeding, interspecific breeding, hybrid breeding like techniques. We just try to manipulate the breeding methods of animals, but we don't alter any type of genes. Uh, we never went for the cellular level. Just let them breed, and we select the animals. So that's a selective breeding. Now here we can further uh, going for our several steps forward to change the gene level. So first thing we discuss polyploidy. Polyploidy, and second thing induced mutation. Last thing is genetic modification. Right. So we discussed already polyploidy, especially in plant, and induced mutation again in plants mostly. But this technique, genetic modification, we can apply for both plants and animal. And this we're going to learn in bigger way in chapter in unit 7 molecular biology. This is a separate section, gene technology and biotechnology, gene technology, genetic engineering, all these field address this genetic modification, genetically modified organism, GMO. So don't worry, this we're going to learn in a bigger way. And last time we discussed the polyploidy. In this method, colchicine is a chemical used uh, to inhibit chromosome separation during anaphase. So it inhibits the microtubule formation. As a result, chromosomes cannot separate into two cells. So we have seen in mitosis, when chromosomes are attached, with the microtubules or spindles. This is the spindle, and this is the chromosome. From this point, centromere, what is going to happen as a result of pulling to these two ends? It's going to separate in the middle, centromere. Then chromosomes can move to the both sides or poles, and then chromosome divide into two cells. But this process is inhibited by the chemical colchicine. As a result, uh, we can produce cells, not just 2N. We can produce cells with 4N, uh, tetra, hexa, octa. So we can even try a triploid, like many chromosomes we can have in one cells. So this is the polyploidy technique we used to produce some better variety of crops using this polyploidy technique. So we discussed this one, and as a result, we have this gigas effect, the bigger size. Because when cells are packed with many chromosomes, cells get bigger and organs get bigger. So we can get a bigger crops, bigger vegetables, bigger fruits like things if we apply this polyploid technique, right? So today we're going to learn the second one is a mutation. Now, mutation is induced mutation. So we have learned previously a little bit about mutation. Mutation we're going to learn in next unit. But mutation is something important throughout the lessons. We learn it is random change of the nucleotide or base of the DNA molecule. That's a natural process. During DNA replication, there is a mistake, there is an error. As a result, a wrong base can come into the place. That's a mutation that happened in naturally. But people realized this can be induced as a result of some kind of experience from uh, radiations and some chemicals. They can also change the base sequence. If, right?
Yes.
Then we can use this gamma rays, protons, neutrons, alpha, beta, and chemical like sodium azide and methanosulfonate. So this is the chemicals we can use to induce mutation, and we can use radiations to induce mutation. Now, this mutation is a big story. Unfortunately, we haven't learned it yet. So, generally, what is important of mutation in natural selection, it can bring new changes to the DNA molecules. So when nucleotide change, the sequence change. When sequence change, codes change. When the code changes, protein change. When the protein changes, phenotype change. So evolutionary, this is important to keep on going. It's just like updating the system. So these mutations can update the system and keep some kind of a different variations ready for any kind of unfavorable conditions existing around. The organism can still face because of these new changes. So that's the important thing of the mutation. Unfortunately, if a person or organism or plant exposed to ionizing radiation or chemicals, this can also induce mutation. Now, this phenomenon is used uh, in breeding techniques. They just exposed to different radiation levels, and after that, you're going to check uh, is my line clear?
Yes.
Because at the end of the month, the data running out, like not sure that. So that is another type of technique used to produce different variations. That means it is happening naturally, but people trying to induce it randomly if they can generate a better organisms. And beyond that, uh, they're going to use cosmic radiation to induce mutation called space mutagenesis. Now, mutagenesis is process of mutations, inducing mutation. So this is really interesting. They take plant seeds to the space, and then space has so much of cosmic radiations. So radiation from cosmic rays is used to speed up the evolutionary process in plants. Then they return these seeds to the earth and then regrow the seeds to see whether they have some kind of advantage. Now, this doesn't guarantee it is always make the better versions. It could also make a worse version, but there's a chance of having a better versions. So all these efforts are done to handle the food shortage, food scarcity, and to the face next level if the population increase and climate change is getting worse. How can we make a better food for the growing population? Then third one and the last one is the genetic modification. I think genetic modification we're going to learn in a separate section. It is included here in a small level. Uh, we can leave it out and we're going to learn it completely from the beginning, how we can modify the genes of an organism to make it better. So if we want to get a still some idea how we can do that, I can give this example, a successful example of this one, right? We take rice plant. Now, rice is staple food of many countries, especially Asian and African and some other countries. It is a staple food. The advantage is uh, rice growing well in these regions and it is cheaper. So many people can afford rice. And it is mainly consists of carbohydrate. Rice has mainly carbohydrate starch. And many years back, people realized developing countries, people rely on eating rice, but they cannot afford other nutrients like proteins, vitamins, and some other essential nutrients. As a result, they develop, especially children develop problems, malnutrition, deficiencies. One of the condition is blindness, poor vision due to lack of vitamin A. And doctors realize this problem is lack of vitamin A. To handle this problem, scientists came up with idea if they can find a gene which make the vitamin A. So this is a gene which can make vitamin A. And this gene transfer to rice plant. Then the new genes are coming to the rice genes. Then it produces vitamin A. So if organisms, this is a cell, and they have these rice vitamin A genes, and there could be another organism. So they also have the genes responsible to make vitamin A. So they try to remove the responsible gene for this. And this gene insert into the rice plant. Then the new genes in the rice plant. Now rice become genetically modified organism because it has different genes. Then advantage is the rice can also produce now vitamin A. So when people consume rice, they also get vitamin A. And this project was successful, and it is called now golden rice because of the color. The yellow color rice is called golden rice. This is one example of genetic modification, but we have a completely committed, dedicated section for genetic modification of organisms. So we're going to learn how we can take this gene, how we can identify the gene, how we can transfer the gene, how we can check this is successful or not. All these things we're going to learn, right? So let's keep it shorter. This principles of breeding method, and it uh, directly change the genes. Okay, last topic of this section. Our aim is to improve the quality, improve the quantity, and provide the satisfaction of the users, reduce the toxicities, and increase the digestion of the food by making the breeding techniques. So we all try to do for the better side. But when we try to do this thing to make it successful, there is a disadvantage coming back. There are drawbacks, there are disadvantages. So we're going to see briefly what is the disadvantage and advantage of this breeding technique. So we're going to alter the natural process. One can argue it is bad, whatever we get benefit for a short term, changing the natural system might have a bad effect in the future. But unfortunately, we have to answer the question for short term. We have to handle the problem, providing quality food, providing the higher quantity of food. Even this genetic modification, so far we don't know what is the long-term effect. So these techniques used some years back and they produce food. So if we want to see a long-term effect, at least we have to wait 50, 70 years. That's a lifespan of a person. So here we're going to briefly check what would be the disadvantage. Now, one of the problem is if we trying to manipulate breeding, we're going to select organisms with desired characters, and we're going to breed them within a small population. So new population is growing from a small population. So there's no new alleles are adding to the populations, because if we try to develop a new breed or new variety, we largely restricted to this first parental breeding, and these are the offspring, and then breeding can happen between parents and offspring, and between offspring. So then populations are growing within a limited mix of alleles. So there's no chance of new alleles coming into this population. So what is the genetic level problem? So mostly it produced the homozygous genotype. It could be AA or simple A, simple A. In genetics, the best genotype is heterozygous genotype. If we want to check the variation within a population, we can use the heterozygosity levels. So heterozygosity level is higher, variation is higher. But most of the breeding techniques produce homozygous genotype. And variations are getting less and less. And there's no new alleles adding to these populations. So it becomes more uniform, homozygous populations. As a result, if there is a environmental pressure, example pathogen, disease, if there's a disease come, and the disease is going to affect almost all individuals, because everybody looks same, everything every plant, every animal has some kind of a same genotypes, not much differences. As a result, if one is infected, we can assume this can spread to all the populations. It's very difficult to control. I think one common example is in the poultry industry. They make different breeds like broilers. So this is again used the breeding technique for a short term period, like 45 days, and they have these breeds, and the breeds have a very again uniform genotypes. And we know well, if there is a disease come to the farm, they have to mostly close whole farm, because they have to completely sterilize everything, clean it up for the next session, because it's going to spread very fastly among all the chickens. That's disadvantage, because they have a homozygous genotypes and less variations, and genetic diversity is reduced. So natural selection is trying to produce more variations, but we are trying to stop that one We're going to reduce variation. We're going to make uniform a group of organisms, and they have a homozygous genotype. So this is considered as very importantly, and we call this actually the inbreeding. So inbreeding is breeding happen within a small population. Breeding happen within a small population. As a result, homozygosity increase. Homozygosity level is high. Homozygosity level is high mean uh, they are less, they are weak in genetics. They have a weak genetic background. So this weakness is called inbreeding depression. This is a big problem uh, in the breeding experiment, inbreeding depressions. And also, if there is a recessive mutation, so bad mutation happen and there is in a recessive form, as we discussed, the homozygosity can increase. Homozygosity can increase both dominant allele. Homozygosity can increase both recessive alleles. So then within a population, this mutation rate is high because next generation, they can also get similar homozygous allele. Within the population, the bad gene spreading is also high. So these are the biggest disadvantage of select artificial breeding. And certain phenotype are not desirable also for the organisms. For example, these are the three examples. We have done selective artificial breeding, but these dogs, like boxer or bulldogs, they have a different proportion of the skull and the upper and lower jaw. So the upper jaw and lower jaw has different size. They have problem of eating. So it's a little bit difficult for them to eat. And when we make sheep, this called Texel sheep, due to a hybridization or mix with different breeds, they have a problem of delivering the offspring because birth canal is smaller and it's very difficult to breed. And even the cows, so they have again very difficult to give birth to the calves. And then we have to do other surgical method to take these calves. These are the negative aspect we have to face when we do for artificial or selective breedings. So this Cattle is used for the muscle, the meat purpose as a Belgian cattle, and they have always problem with deliveries. So they are unable to deliver the calves. So then surgical method need to take the calves. Same with this Texel sheep and boxer dogs. They have eating problems. So these are the negative aspect of selective and artificial breeding. All right, so we have completed the unit 6, genetics and breeding. I suggest if you have a time, again, go through these lessons, videos, because more we can remember, it's better to handle the issues, I think, compared to the other units. Genetic is very special because we have a question to solve using logical method. Other areas we don't have this kind of extreme logical analysis, but we have seen when we trying to when you're trying to handle questions of breeding genetics, they are very logical type of questions. So we have to analyze carefully to find the answers. Okay, do you have any other questions about this unit?
So I did the 22 papers. I have small doubts in that.
You mean which genetic questions?
Yes.
Uh, did, didn't we have this question before? We have done it. I mean, how many questions you did?
22 paper. I did some questions in that paper. I have small doubts in that question. One of the questions.
Do you have it?
Yes. Uh, so in that 34th question, a point mutation.
Okay, let me get this question. Okay, which one? 34.
33 and 31st.
34. Okay, a point mutation. Anyway,